Today's topic, as you can see, I have written it already on board. This is about gene cloning. Before I enter to the main topic, today's focus will be on gene cloning. Let me give you a brief introduction about the artificial cloning methods. There are various other methods also which is oriented with this cloning. The purpose of cloning is to produce multiple identical copies of gene-sized pieces of DNA molecules. Now this has a it has a large field to be executed in the field of molecular biology. Today's focus will be on gene cloning and molecular cloning we will be concentrating on. Other than this, we also have reproductive cloning, therapeutic cloning. I'll come to it little bit in this video. In my later videos, you will be finding details about reproductive cloning and therapeutic cloning. So gene cloning is primarily about producing identical copies of gene-sized DNA molecules. The steps that are initiated, actually cloning, molecular cloning is actually about a set of experimental methods. It has parts of experimental methods which are executed in the field of molecular biology. Thus, we can have recombinant DNA molecules as a mechanism of gene cloning. Let's see what the mechanism of gene cloning has in store for us. We know that in molecular cloning, we are going to prepare identical copies of gene-sized DNA molecules, and that gene that will be the target gene or the gene of our interest is going to be inserted into a host organism, and this host organism is going to reproduce and give us more number of that target gene which is going to encode a protein. So this is the basic idea. So let's see, this is the foreign gene. Here is the foreign gene, means this is the total genetic material I am showing you. The target DNA is somewhere here which has to be taken. Now I do not need the whole of the genetic material, I need a certain part. So what is going to be done, as we know that in DNA recombinant technology, our main thing is the activities are cut, copy and paste. So here a restriction enzyme is going to cleave. Say here the restriction enzyme, digestion of restriction enzyme will be taking place. That is the digestion of the target gene is going to take place. That is with the help of restriction enzyme. Now what is the restriction endonuclease that will be used? Let's come to it little later. And this, after being cleaved, it has to be inserted. So it cannot be directly inserted. We need something to carry this foreign gene, and that carrier is none other than a vector. This is a plasmid, a cloning vector we have chosen. This plasmid vector will help us to carry this foreign gene, and it will be inserted into this bacterial cell which is our host. Now bacterial cell also has its own chromosome, and this plasmid will be placed here. This is the basic idea. Now how, the first point that is isolation of the foreign DNA, and after being isolated it will be transferred into this vector. Now which restriction endonuclease I am using? In this case, say I am using BAMH1. If I am using BAMH1 to cleave this DNA and to be placed, naturally here also in this vector, in this plasmid vector which will help in cloning process, the same restriction endonuclease has to be used. What is the reason? The reason is a basic compatibility has to be there. The same sticky ends, remember that the restriction endonuclease that will be used for cleaving, it has its own recognition signal. So the signal sequence has to be same. If the signal sequence differs, then there is a problem in getting the same sticky ends. And if there is problem in getting the same sticky ends, it will not be ligated properly. So our motto will not. BAMH1 is used to cleave. Here in this vector, in this plasmid vector, the same restriction endonuclease has to cleave, and that is BAM-H1. If this is like, this is our PBR 322. If you remember, I have already discussed in my video tutorial about BR 322 plasmid vector, the cloning vector. If you have missed out, please go and watch that video. So here I am just going to touch up little bit that in BAM restriction endonuclease is that is used is BAM-H1. In PBR 322 also the same restriction endonuclease will be used, and this is the site for the BAM-H1. I am not going into other names of other restriction endonuclease. What are the names? BAM-H1, SAL1, EcoR1, CLAV1. These names, HIND3, I am not using it right now. Right now, I am just focusing on, if I am using BAM-H1 here, then BAM-H1 has to be used here also. And the cleavage will, that means opening BAM-H1 is used to open the plasmid vector at this site, because BAM-H1 is active at this place only. If there is incompatibility, problems will arise. Now, as BAM-H1 is opening this plasmid vector, due to the process that this foreign gene is going to be inserted into the plasmid vector, very important sentence, as the foreign gene that is isolated is going to be inserted at the site where the BAM-H1 is to cleave the plasmid vector. Due to this, it breaks. What is the reason the plasmid vector breaks at this point? The reason is that here it loses, we know here tetracycline is going to be, the gene present at this region. I'll use some other color. So let me use this green color. This is the place where the gene encoding resistance to tetracycline is present. As BAM-H1 is cleaving at this point, is going to break the point because already the gene encoding resistance to tetracycline is present over there. So naturally it will break. This is the place where tetracycline resistance gene is present. TET are in this way you write. This is the gene that is present in resistance to tetracycline. So BAM-H1 is used at this point and naturally it breaks. Now the foreign DNA has been inserted into this vector. Now what has happened? Here the foreign DNA is inserted into this vector. So it has become recombinant. Why it has become recombinant? It has become recombinant because foreign DNA is inserted into the vector. If it was not inserted, it was a non-recombinant. In case of a non-recombinant, the foreign DNA is not inserted. Now, how we understand recombinant and which is a non-recombinant? I have said that recombinants will carry the foreign DNA inserted into it. As it has happened in this case, here it has been the foreign DNA which has been cleaved with BAM-H1. Here BAM-H1, the plasmid vector is our cloning vector, which is the carrier for this foreign gene. It has been inserted. Why? Because it is BAM-H1 has lost its, the genes that are present over there has lost its resistance to tetracycline, so it has broken up and it has been, the foreign DNA has been inserted. Let me write over here that foreign DNA is already inserted in this case, and this is going to be now placed into the bacterial cell. That is, the plasmid will be placed in this bacterial cell. This is the bacterial cell which is carrying the foreign gene in it. So this is a recombinant. We know this. Okay. This is the recombinant molecule of DNA, and this is going to carry that foreign gene in it. Now in this case, one confusion may arise. There are many bacteria. Suppose these bacteria are going to be placed all into a culture media. Now, which bacteria has got that foreign gene and which bacteria doesn't carry that foreign gene? That is a big confusion in front of us. We know that we have inserted, but whether they have taken or not, that condition while culturing, whether it has happened or not, that is a big confusion, and we do not want a confusion. Cloning is a process, tedious enough, time consuming, expensive, so we cannot run the risk. We need to be clear with this point, whether it has the recombinant DNA in it or not. So there is a trick. Let's understand this trick. Say this is a place where we have a certain media, a buffer solution always required to maintain the conditions of pH, and T4 DNA ligase we have used. The reason of using ligase is so that it joins. Now I have placed the DNA molecules and the plasmids. Now what happens is, these are the plasmids which have been cleaved, and these are the foreign DNAs which have been cleaved. Now all of them along with ligase have been put. Now what are the three consequences? Consequence number one is, let's see the consequence number one, result. What is the result of this experiment? It may happen that some of them will be recombinant, that is carrying the foreign gene, which is our motto. Our motto is that they will carry the foreign gene. So this will happen. Okay, it has happened, very good. That is our motive of the experiment. But this is also a greater possibility that some of them will self-anneal, self-ligate. Sorry, not self-anneal, self-ligate. Because these plasmids have got the opportunity, they combine with their own selves. And this second possibility is self-ligation may take place. And we get few plasmids which are self-ligated, which do not carry the foreign gene. So these are non-recombinants. And some will have only DNA overhanging, like these DNA molecules also join, self-ligate. So that may also happen, that self-ligated plasmids may take place, self-ligated DNA molecules also take place. These are the consequences. So we need to be very careful also. So now the next step, the first step was isolation of the foreign DNA. Then it has to be inserted into some vector using the same restriction enzyme, and then this vector is going to be placed into the bacterial cell. Now one point that we have to remember in this case is that these bacterial cells require suitable conditions also for their reproduction. The foreign DNA are the recombinants, and the plasmids which do not carry the foreign DNA are the non-recombinants. Now we know that we have to use selectable markers. Why we need to use the selectable markers? The use of selectable markers, now this time I am rubbing this place so that I can use this place. Let's see what is the utility of using a selectable marker. Selectable marker is actually selectable markers. Select, these are large nucleotide sequences, not very large of course, medium sized, selectable markers having some nucleotide sequences and having resistance to some antibiotic genes. So selectable markers are used to differentiate which is the recombinant and which is the non-recombinant. So in this case, the recombinants will be carrying the foreign gene, we know this fact, and the non-recombinants are not carrying the foreign gene, and that work will be given to the selectable markers. So selectable markers, in this case, we will be using ampicillin, as these will have resistance to ampicillin. The genes are present in the genes which are having resistance to ampicillin and tetracycline. That means the two antibiotics that can be used as selectable markers are present in genes which are having resistance to ampicillin and tetracycline. Now the question is, I have mentioned about BAMH1. BAMH1 is present at the site where the gene has resistance to tetracycline. It doesn't have resistance to, these have lost the resistance to tetracycline. Why these have lost resistance to tetracycline? Because I have inserted the foreign DNA into it. As the foreign DNA has been inserted into the site where BAMH1 has cleaved, so the genes have lost resistance to tetracycline. Naturally, the concept says that if I am going to plate it in a medium where there is tetracycline, it will not grow. So these are non-recombinants. So that means here the concept has to be clear, which is the recombinant and which is the non-recombinant. For that purpose, we use a media X-gal. So let's see what the trick is. It's a screening process we will be doing for recombinants and non-recombinants to distinguish them using selectable markers. And what are the selectable markers we will be using? Ampicillin and tetracycline. If the bacteria is not carrying the gene which is having resistance to such antibiotic, then how it will survive? It will not survive. That is the concept. But let's see how the concept has to be executed. Over here, now that we are going to use selectable markers to differentiate between the recombinant and non-recombinants, let's see what the process seems like. I'm rubbing this part so that I can write it. I'm not rubbing the main portion, so that always you can keep in mind exactly what is happening in this cloning process, which are going to help us in producing identical copies of genes. Now see, recombinant and non-recombinant, that is the basic purpose over here. Now say I am placing, this is a plate for culture, and there is a certain media that is used. I have a choice of X-gal also in this case, and this media is filled with the genes that are having resistance to ampicillin, this antibiotic, and the plasmids are going to be placed here. These are the plasmids which are placed. Let me use another color so that distinguishing, this is the medium, and here plasmids are transferred. Remember this sentence, plasmids are transferred into a medium where the antibiotic resistance genes ampicillin, which is having resistance to ampicillin, is present. Now the plasmids are placed in this media. So that means if these plasmids are being placed into a media where there is genes having resistance to ampicillin, not tetracycline, what is the result? The result is all of them will grow. Why all of them will grow? Because here there is no question of tetracycline. So all of them, whether it is recombinant or it is non-recombinant, both have the capacity of growth. Why? Because these genes are having, it is placed into a medium where the genes having presence of resistance to ampicillin is present. So there is no question of any botherance over here. Whether it is recombinant or non-recombinant, all will grow. But at the same time, if simultaneous plating should be there, at the same time I am having another plate. In this case, I am having genes which are having resistance to tetracycline. Now here comes the medium, the media that is there used is having antibiotic resistance to tetracycline. Sorry, it's not ampicillin, it is tetracycline in this case. So it's going to be TET, and here I am going to place the plasmids. What are the consequences? When I am placing the plasmids into a medium where the genes which are having a resistance to ampicillin is present, all of them show growth, whether recombinant or non-recombinant. That is, whether it is carrying the foreign DNA or not, it is going to show growth. But the same thing, that is all the plasmids now I will of course not take all the plasmids together. I will take a set of plasmids again, and this time the medium where it is placed, here there is antibiotic resistance to tetracycline, not ampicillin. What is the result? The result is that those which are recombinants, remember the recombinant is going to carry the foreign gene and it has lost its resistance to tetracycline. So here in this case, the recombinants will not grow and the non-recombinants will grow. So whichever is our recombinant, let's mark it like this. This is a recombinant. This is also a recombinant. This is also a recombinant. So these will show growth or not? Can you answer it? Now it's very simple to understand. As these recombinants, what is the reason? The reason is the recombinant genes have lost their resistance to tetracycline. So when they are placed into a medium where there is tetracycline is present, no growth. But at the same time, where the non-recombinants are there, these will be based on this that is screening process. Right now I have not used any chromogenic substance as such. Still now I have not used, a simple medium I have used. So here the process isolation of the foreign DNA, then placing it into a plasmid vector. The vector is going to carry that into the bacterial cell, and when the foreign DNA is transferred into the bacterial cell, the process is known as transformation. What is the process known as? Transformation. When I am placing the plasmids into a medium where the genes which are having a resistance to ampicillin is present, all of them show growth, whether recombinant or non-recombinant. That is, whether it is carrying the foreign DNA or not, it is going to show growth. But the same thing, that is all the plasmids now I will of course not take all the plasmids together. I will take a set of plasmids again, and this time the medium where it is placed, here there is antibiotic resistance to tetracycline, not ampicillin. What is the result? The result is that those which are recombinants, remember the recombinant is going to carry the foreign gene and it has lost its resistance to tetracycline. So here in this case, the recombinants will not grow and the non-recombinants will grow.